A very good morning to my class. This is chapter 23, Evolution of Populations from BIO 101. This chapter follows the chapter 22 where we discussed descent with modification and Darwin's theory of natural selection. Okay, so by this time we are all in complete agreement on what Darwin's theory was all about, and the evidences that he provided was strong to convince everybody. But this chapter mainly talks about how it is not the individual that evolves, it is the population that evolves. So for that, basically it's the individual who accumulates a certain set of traits, and if the traits that this specific individual accumulates is beneficial for survival in the circumstances or in the kind of environment that they live in, you would have more number of individuals being introduced into that environment as forms of offsprings of this first individual, and slowly over a period of time, over a period of generations, you will see that the entire population will have acquired that specific quality, okay, or that trait. So, in this case, the first example, the simplest one, the way we would explain is if you have a dark kind of environment and there are many beetles present in it, these beetles can have different color shades with which they are born with. Okay, so there are light colored beetles, okay, and there are slightly darkened beetles, there are absolutely dark beetles. All right, so all of these exist in this population, so that means they have varied traits, inherited traits. A finch or a swallow, when it comes in search of something to eat, can easily spot the white or the lighter colored beetles against this dark background. This dark background is the environment in which they are present. All right, that is the condition that they are existing in. And so the individuals with certain traits get eliminated because they get eaten, they get spotted, and they can't escape, and the survivors reproduce, and the survivors end up having babies with their qualities, which means more and more of the individuals in the population will have the darker shades. This means there is an increased frequency of the traits that enhance the survival and reproductive success of that particular individual in that population. Okay, so another example, this is in real life. Prior to the chemical synthesis of DDT, all right, the pesticide DDT, in the wild population of the pests that they had looked at, not all of them would be resistant to this particular pesticide. Now, I'm taking the example of DDT, and the entire population, probably a few of them were resistant to DDT. And the population had a varied set of these pests which had varied sensitive ranges, sensitive or resistant. With the application of the pesticide and how do I say, more frequent application of this pesticide, many died, yes, but some survived. And the ones that survived were resistant to DDT, which means they passed their genes to the future generation of these pests. Almost all of them will end up having resistance to that DDT. Okay, so future applications of this pesticide will become less effective, and then the frequency of these resistant pests will increase in the population. So, mutations and adaptations occur at an individual level. However, the evolution occurs at the population level. Okay. So this is an interesting picture that I would like to add. This is found apparently in the deserts of Algeria, where they found these goats were capable of climbing on into these thorny trees. So they're all up there. This is no magic, this is no photoshopping, and this is really true. And what I would like to show in this is first talk about a little bit about goats. Goats are kind of survivors. They will eat just about anything. All right. So even if the environment is absolutely arid and dry, they will survive by eating any vegetation that they can get. And in this area, the lowland grasses are practically nil. So the thorny trees are the only options for vegetations, and some of these goats have developed the ability to climb and are able to balance themselves on these thin, all four legs on these thin branches. That means these are individuals which have a certain quality of being able to balance themselves. But over a period of generations, when you go by, if this is the same kind of environment that still continues to be, you will see almost all of them will end up having this quality because they are the ones who have survived, right? So right now the condition is like this, but who knows what's going to happen in the future. Okay, so there is also another term that you need to know, microevolution. Microevolution is evolutionary change in a population in the smallest scale possible. Okay, and that means there is a small change that you can see, but if you have accumulation of such small changes over a period of time, it can lead to a complete change in the population, and they would completely differ from a neighboring population which probably were arising from a common ancestor, the main land, and they would be completely different. Okay, so for this I would like to also use the story of the finches in the Galapagos Islands, the relationship between the soft fruits and the beaks, or drought and hard seeds and the beaks in Galapagos Island. Okay, so here you have finches with different kinds of beak types. Okay, so a cactus eater has something that with which he can gouge out soft fleshy fruits. A seed eater specifically has the ability to break open the hard nut. An insect eater has got a pointed kind of beak who can go through the grooves and access the insects from in between the bark. These are directly connected to the kind of environment and food source in the islands that they are present in. Please note, not all of them are present in all the islands. Okay. Now, another point to remember is there are some variations that are not heritable, meaning you do not inherit these variations, these traits, but it depends on the diet. Believe it or not, this particular caterpillar looks like the flowers of the oak tree by having consumed the flowers of the oak tree. Vice versa, the same caterpillar looks like the twigs of the oak tree by consuming the twigs and the leaves of the oak tree. So this kind of variation depends on the diet of that particular individual creature in that population. Okay, so they owe their appearance to the chemicals in their diet and not their genotype. Okay, those raised on oak flowers resemble the flowers. Those raised on the oak leaves resemble the oak twigs. Now, in other cases, variations can occur. Sometimes inside a population, you will see lots of variations. Sometimes between populations, you will see lots of variations. And sometimes you will see like a gradual change in a specific quality of a population depending on its geographical presence. Okay, we'll discuss each one of these examples. So, in a population of snails, you will have varied kinds of shell colors and stripes present in them, and also sizes. So this directly compares to people of different skin shades and heights present in the human population. Okay, so within a population you can have variation. Now, between populations, variations. Now, this actually goes back to a story which is listed out in your textbook. It's actually a fact, but I'm relating it in the form of a story. So, there is this island, and a boat full of explorers landed here at this coast. And from this ship, okay, the mice kind of got offloaded in this island. All right, all through they kind of spread all over in these parts of this island, and this is what that population of mice seem to have in their karyotyped genes, chromosomes. All right, a few of them managed to kind of move, and this was like the open plains in the coastal areas. This is the mountainous areas of the other geographical region in that particular island. They kind of moved here, and when they moved, they kind of stayed in these regions. Not all of them had ever a chance to go back. Now, open plain versus a mountainous region, there might be different demands of this environment, and mice with different kinds of traits may have died, or beneficial kind of traits may have lived. So, depending on that, certain genes may have completely been transmitted to the entire population of future generations. And then when you compare these mice, like after say 50 years, which is what they did, 50, 60 years, they kind of took the mice and their genetic makeup, they studied of the mice in the mountainous area and in the open plain area. And they kind of differed in different chromosomes in many ways. This told you that though they arose from the single same population as they had been offloaded, over a period of time, because of certain environmental changes and the demands made for survival, certain mice with certain traits got selected, and so their qualities were the hand-me-down for future generations. Right? So you can see variations between population. And to talk about the cline that we were talking about some time back, in 2-3 slides back, I need to give you the example of this particular fish. Okay, this fish is found in cold waters of Maine and warm waters of Georgia in the eastern coast. Now, if it is cold, which means how are the enzymes in the body of this fish going to work in order for them to be able to move faster and burn energy so that they are able to make energy to make their muscles move? We are just taking the example of one enzyme. All right, the enzyme that I am dealing with is lactate dehydrogenase. Okay, this lactate dehydrogenase is present in this fish, present in this cold areas, present in this fish, present in this warm areas too. But the ones that are present in the cold areas seem to prefer the cold. The same enzyme type is not present in the fish or in the population of fish in the warm areas. So they kind of looked for the cold preferring allele over the population of fishes along the coast, and they found more of it concentrated up the north. And as and when the latitude came towards the tropics or in the warmer waters, the number of fish having that particular trait is kind of sort of less. They exist because you see there is nothing separating these fishes except the temperature. So it's like the colder water preferring fish stay, and a lot more of those fishes have this specific cold temperature preferring enzyme gene versus in the warm. So they are present in a kind of gradation or a cline. And I hope that's understood. Okay. Then we move on to the part where we are going to talk about what are the different sources of genetic variation. Okay. So the first thing, if you remember your chapter on meiosis, okay, so sexual reproduction, okay, that involves meiosis, and in meiosis you have two spots or two particular sequences where you have genetic exchange and variation being created because of random kind of independent assortment. All right. So crossing over an independent assortment, this occurs during prophase 1 and this occurs during metaphase 1. These are the two spots wherein you can have a lot more variations introduced. The third part is random fertilization, meaning the male that is present and the female that is present, you do not know what is the genetic composition that this female is got and what is the composition the male has got, genetic. So if they with complete variations come together, they are the ones introducing more variation. Okay, this goes to the fact that you kind of marry far away from the family and not closer to the family. That's an important thing to remember. Random fertilization. So these are the parts where you will have variation being introduced. But that is a finite number of variation that you can get. Okay. Mutations are your best bet for changes because this is one place where you don't know where your UV is hitting, in what part of the DNA and what kind of changes it is offering to make. Mutations should not be considered as all bad. Mutations should not be considered as all good either. Most of the time, if there is a mutation, there is a change. That change might be good, useful, helpful in survival, in which case a lot more of the population, future population, will end up getting that changed genome, and that kind of gets introduced into the population. So this is a new variation introduced, and that is what has changed us from the lowly single-celled microbe that existed in the pool of the seas millions of years ago, and we have mutated over a period of time and become the human beings that we are now. Okay. Now, one more important thing to note is it is very difficult to observe this kind of mutations and the effect on a population in humans, but it is easier to observe that in bacteria. Why? Because of their ability to produce, reproduce, because of their short generation time, we are able to see the change in the population over generations that have come by after a certain exposure to UV, and we can see what effect it has had on the population, and you can see the variations being introduced in that. Now I move on to talk about, this is New York, okay, in the 1876. This is New York in 1932. This is New York in 1988, and this is New York in 2013. Now you can see very clearly there have been changes introduced. So when can you see and state that there have been changes? When you can compare. So you see what was there in the past and you compare it with the present, this present, and then you can talk about what are the changes. How will we be able to do that with the past that has gone by? It is a little difficult for us to know. What you can do is to check the kind of gene frequencies that existed then and the gene frequencies of a certain particular gene that exists now, compare it, and you kind of get the idea. Is it the same? Has it changed? Has the entire population evolved or not? Alright, so for this, it is not very easy to do like we can look at a picture and do it. There is a certain mathematical equation introduced by two scientists. They are Hardy and Weinberg. So remember, there is variations present. Just because that they are there, there is no guarantee that a population will evolve. For that, you should see that at least one of the 5 factors must be at work. Only then you can say that the population is evolving or changing. If all of those 5 factors are constant, then you can very happily say the population is not evolving. Also remember, if you are talking about a population, like some time back in the beginning slides, I talked to you about a certain pest. Okay, so this particular pest, all right, looks the same that it did so many years back, and yet I can say that the population has evolved to have the resistance gene. So I come back and reword my sentences. Evolution of a population does not mean a complete change in the appearance of that population. It can be that certain genes of that particular population, present in that particular population, in the individuals, might be different now than what it was. So certain genes, there might be no change at all. Certain genes, there might be a change. So you will still have the conclusion, yes, the population is evolving for that particular quality. Right? Let's see what is the test that we are going to talk about before we assume, presume, or talk about evolution. Okay, so Hardy and Weinberg were two scientists. They were mathematicians. They kind of said that a population is at genetic equilibrium and not evolving if the allele frequencies are not changing. Okay, what these words mean, hold a little bit. In the next slide, I'll be talking about it. So, allele frequencies are not changing, they are the same, means the population is not evolving. When will this happen? If there is no mutation, if there is a large population, if the population is very stable, there is no immigration or immigration, meaning there is no one coming in or there is no one going out, and mating is random. And Also, there is no natural selection happening. Meaning, all of these conditions, if this is the way they are, then all of the allele frequencies will be unchanged. And then you can say the population is not evolving. And then you can say you have Hardy-Weinberg's genetic equilibrium present. Okay, let's talk a little more in the next slide. Okay, so first thing what I would like, what I do in my class is I kind of say, okay, these are the number of students in my class. See, there is 17 or 20 or 25 students sitting in my class. So this is what I would say. Everybody has got 2 alleles for every trait or every character. Sorry. Everybody has got 2 alleles for every character. For example, eye color. There will be 2 alleles for that. One given by your dad, one given by your mom. Which means if there are 25 students in my class, there is 50 alleles in my box of gene pool for eye color. Does that make sense? Okay. So, a gene pool is all the genes in a population. And it is like a genetic resource that is shared in theory by all members of the population. Meaning it is just there in a box, anybody can access it. In theory, okay? Because that's not normally, we'll come to the understanding later that that's not normally possible. But right now, in theory, let's presume it is just in a box. All of the eye color alleles are in a box. Right? And that is the gene pool for that. For each gene, there may be 2 or more alleles, depending. Supposing I'm talking about, say, blood group, there might be more than that being added into the gene pool. Alright? And individuals inherit different allele combinations. Okay, so now I'm going to talk about an example of 500 wildflowers and flower color. The gene for flower color. 500 wildflowers. So, the gene for flower color, with that, I have 320 red flowers. That means they are red because they have 2 alleles present in them as CR. They have 100 and just a second, where did it go? Okay, 100 and I remember they have 20 white flowers, which means it is CW. Okay, and I have 160 pink flowers, which is got alleles as CR. Okay, so now if there are 2 CRs, means what is the total number of CR alleles that are present in this entire box full of these 500 flowers? First remember, if there are 500 wildflowers and flower color, means there is 1000 alleles for flower color totally, because each flower will have 2. So CR alleles, how many do I have out of this 320? Means 320 of into 2, which is 640. Okay, and then in the pink one, because it has CR and CW, it has plus 160. That is here. Let me put it here. 160 from the pink flowers. Okay, which is 800 CR alleles are present out of 1000 alleles that are present in the box Okay, allele population that is there, which means what is the percentage that they are present in, or their allele frequency is 0.8 for CR. Okay, so allele frequency P is 0.8. All right. Now, on the same note, for CW, okay, you have 160 from this, all right? So that's 160, and CW is 20 into 2, which is 40, which is 40 here, which makes it 200, and 200 CW alleles out of the total 1000 makes it 0.2, the allele frequency. So the allele frequency Q is 0.2. Okay, so this is what we do when we are discussing the total number of alleles present in this population. If out of 500 flowers, if we put all of those alleles in this box, assuming in theory that they are all equally accessible, this is how you calculate what is the allele frequency, and you kind of name each one of them as the small p or small q. Okay, so this is the first step. Now, Hardy and Weinberg came up with an equation to talk about allele frequencies and said, if this is the way you find out the allele frequency, then, and they are present in this gene pool and all of those 5 conditions are met with, that is when you can say that they follow this particular equation, okay, which is, one second, let me have some space to write this, and let me see if I can write that up. Okay, so he calls that p square plus 2pq plus q square equals to 1. This is your Hardy-Weinberg equation. He says you will have allele frequencies. They will keep shuffling, following the rules of Mendelian principles of segregation. Principles of segregation. Shuffle, and you will keep having them continuously being present in that same number in the population, as long as there is no mutation, no natural selection, the population is large, there is no immigration, nothing coming in or nothing going out, no immigration, and there is random mating. Now, this is the time when I tell my students that these 5 factors are not necessarily possible in an natural environment. There is always going to be natural selection. There is always going to be some going in or some coming out. There is never random mating. Now, what I mean by this is, think about a lion. Okay, the strongest lion is able to defeat the other lions, hold on to a territory, have a harem of lionesses to have its babies. Now, the lion that loses, does he get a chance to mate and pass his genes on to the next generation? No. It is selective mating. Because, remember, females don't like losers. Females are smart. They would prefer to have babies with the male who can provide for her and her baby. In all living forms, living systems, if you notice, if she gets a chance to choose, she would choose the best male possible. So mating is not random. Mating is also very, very selective. Towards the later on pages, you will see that it is very, very selective intrasexually and intersexually also. And there is always a bombardment of UV, electromagnetic radiation, and many other chemicals that we are continuously being exposed to. And there is always some mutation or the other that's taking place. So, and there is not necessarily that population is always very large, because though the population is very large, in a certain spot, there might not be enough numbers for it to not play a significant effect. All right. So population may be large overall, and so overall you can calculate that yes, it has not really changed the population much, but micro populations, there might be a greater degree of differences observed in that. So my point is, Hardy-Weinberg said, this is the equation that holds true, and the Mendelian principles of segregation and assortment will keep happening, and the allele frequencies will keep appearing constantly in those numbers and not just disappear if there is no mutation, if there is no natural selection, if the population is large, if there is no immigration or immigration, and if there is random mating. But in real life, mind you, all of these things cannot be presumed to be not taking place. They do take place. So my bottom line is, there is evolution taking place. There is work at play for evolution to be happening at every level. So this is what he does, and I walked you through these slides in the previous slide itself. He is talking about the 500 wildflowers. There are 1000 alleles. Then you have 320 CR, and then 20 CW, and CRCW would be pink, that is 160. And you kind of find out what is the allele frequency for the red allele, what is the allele frequency for the white allele, and you find out that one important thing to note is the sum of all the allele frequency will always be 1. P plus Q will always be 1. Okay, so Hardy-Weinberg rule, he says at genetic equilibrium, the proportions of these phenotypes would always be 1, and you will have these allele frequencies constantly being expressed. Okay, the state that the genotypes will remain constant from generation to generation, provided only Mendelian segregation and recombination are at work. It's like a deck of cards. No matter how many times you shuffle, you cannot change the number of aces. It will still be the same. So he walked you through this, and I did too. So the conditions for Hardy-Weinberg equilibrium is for a non-evolving population, which is rarely met in nature. This is an important sentence. Non-evolving population means there is no mutation, there is random mating, there is no natural selection, it is a large population, there is no gene flow, meaning no immigration and no immigration. Okay. Now we move to the part where we talk about what are the mechanisms and processes that can alter allele frequencies in a population. So there are 3 mechanisms that can change the allele frequencies. Natural selection is one very important one, and it's kind of very slow. It is very much dependent on the environment. Okay, so natural selection is a process where certain inherited characteristics are more likely to survive and reproduce, and the other organisms with other characteristics might not have the chance to increase in number. So this kind of helps in selecting the best possible under the circumstances. On the flip side, genetic drift is kind of deleterious, wherein some harmful genes stay fixed, meaning they are there to stay in the population and you can't do anything about it. And gene flow kind of is exactly the opposite of genetic drift, meaning there is always a chance that you will get fresh from another environment and you can sort of see this calamity through kind of situation, and that will be explained in a few slides down the line. Okay, so for example of natural selection, some slides back I had explained this with a picture, but we are talking about it with words now. The fruit fly Drosophila melanogaster, melanogaster, it has an allele for resistance to insecticides, including DDT. The frequency of this allele was 0% in the lab strains collected from the wild in 1930. Okay, so in the lab they still have strains that they had collected at 1930 and before, and they have 0% frequency for resistance to DDT or any other insecticides. But when they collected samples from the wild after 1960, in the wild they noticed 37% of the population had this particular gene present in it. So that means the population has definitely evolved. They've gotten the Drosophila with the resistance gene has been naturally selected, and so a big chunk of the population has those resistance genes. This is because there has been, first thing is the mutation has occurred between 1930 and 1960, or maybe the allele was present but it was very rare, but it is now on an increase. The reason for which was between 1930 and 60 is where we chemically produced DDT and used it to spray crops. It was abundantly used all over in the farms and reached the water bodies, forests, and in different ecosystems it has managed to reach. Alright, so if the organism which has survived means it is able to overcome the presence of that chemical, that means it has the resistance gene. Okay. Now we talk about genetic drift. Genetic drift, there is two types. The foremost important thing that you have to remember is there is a random change by a chance event. The most important word in this, the population is small. Okay, so population being small, which means it can have two types. One called bottleneck effect, the other called founder effect. Okay, so bottleneck effect, basically you can have severe reduction in population size. One example that they kind of give here is the elephant seal population. Previously there was a lot of them all over in the Arctic coasts, in the northern hemisphere near the colder areas. They have been hunted down to just 20 individuals. And then suddenly people woke up to the fact that they are kind of going to lose this particular population of seals, which is important to the ecosystem there. And they kind of cordoned off that area, made that a protected area for them to continue to thrive, and hunting was kind of banned. Okay, so now the population, after so many years, like 10, 15 years, the population has rebounded 30,000. But remember, all of the genes that these 30,000 seals have come from a hand-me-down of these 20 individuals, which means previously there might have been 1,000, 20,000, 12,000, 14,000, whatever number. They might have had more variations, but all of those variations are gone. What is left is just these 20 individuals. So whatever you have got now, there are many genes. Like in 24 genes, there is no allele variations at all. So those are the only kinds that you have for those 24 genes that they are talking about, which is kind of sad, because more variations you have, more chances for that particular species to survive under an adverse condition, okay, or for resisting a certain disease or adapting to a certain environment. All right. So my point was, for this particular bottleneck effect, original population had, like these marbles, different kinds and in different frequencies they were present. All right. With all of them dying off, this was the only surviving population. Now even if they increase in number, there will be almost all of them which will have this quality and some with this quality, and that's it. The yellow quality is gone. So I hope with this picture it's kind of clear to understand what is the outcome of the bottleneck effect. Another example is when you had famine and it wiped out most of the people who perished during the famine in Ireland. So then there was a very small population that was left behind, which then increased and rebounded in number. But the genes and the variations contained by the people who died is lost forever. They are not carried on. They are not there now in the present generation. Okay. Then you have something known as founder effect. Now, founder effect, I have three examples that I can explain as to what this basically means. Founder effect is when a small number of individuals start a new population. Okay, so the best example that I know closer to what I have experienced is a particular group of ethnic group present in India. They are Zoroastrians who came on 2 boatloads to the west coast of India. Okay, so that is the only number that of people that came. Presume, I'm taking a wild guess, say about 800 people came in these 2 boatloads. They came over, they settled themselves, they kind of made themselves indispensable to the society and the economy in India. But they had 2 very important rules. One, they did not marry outside their community. Did not marry outside their community for a very, very long time. Which means these 800 people were the ones that kept meeting for generations, and they would have the future generations, all the genetic material that was present in these 800 people is what is transmitted down. And because they did not marry outside the community, a lot of recessive genes and a lot of deleterious genes kind of stayed fixed, like for example dementia. Okay, and this is also observed amongst the Amish who do not marry outside the community, in which case there are certain qualities that have become fixed. One example is polydactyly and depression. Okay, another example is the Ashkenazic Jews in Eastern Europe. So Again, they were kind of cut off from the rest of the world till recently, that you have air travel and people moving around all over the world, global travel possible. They were marrying only amongst themselves. So a particular disease called the Tay-Sachs syndrome kind of stayed fixed, in the sense it's very common for people from this community to have children suffering from this particular disorder, because many of them carry this recessive gene, and if two carriers meet, the chances of them having a baby with the both the recessive genes is likelihood is higher. All right, so the point I'm trying to make is, when you have a small number of individuals to start a new population, the allele frequencies of the founders may not be the same as the original population, and so the effect becomes very, very pronounced, especially on isolated islands where they do not have a chance to mix with the main population. Okay, another example that is present in your textbook is Tristan da Cunha Islands. In 1814, 15 British colonists had founded a settlement in this island. This is midway between South Africa and South America. One of the early colonists had a recessive allele for retinitis pigmentosa. It is a progressive form of blindness present in homozygous individuals. Of the founding colonists, original descendants, okay, of 240 descendants in the late 60s, 4 had retinitis pigmentosa. So one person, so that means it was 1 out of 15, but now it has become 8 out of 240. So you can see what I am talking about when you see the that the frequency of this particular allele that causes the disease is 10 times higher on Tristan da Cunha than in the populations from the where the founders came from, right? In the original British colony that they came from. So, genetic drift. What are the effects? A summary. First, it is significant in small populations. It causes allele frequencies to change at random. It can lead to loss of genetic variation. It can cause harmful alleles to become fixed. What this basically means is to stay. They are here to stay. They are there in the chromosomes of the majority of people. That's what the word becomes fixed mean. Gene flow, on the other hand, is the exact opposite. It's the exact opposite of genetic drift. So in genetic drift, if you remember, the problem was, and a population is present and the population is isolated, there is no other mixing with the mainland, and so there are certain genes that kind of stay fixed, and it kind of becomes like a negative effect. Gene flow, on the other hand, basically talks about in and out. So there is always a chance of some individuals with a variation getting into this population and kind of making sure that future generations may have these new variations, possibility of these new variations also, versus some of the existing genes can get out to the mainland and you can have that particular gene also getting mixed up there. So it's basically like a blend that's possible. So gene flow basically means physically flowing out of the alleles into or out of the population. This tends to keep the populations similar. It counters the differences that you would result from mutations, natural selections, and genetic drift. Okay. The example that I have is, there is this mine that is present, okay, a copper mine. So when you have these mines, you will also have this acid wash, acid washings, or okay, or they call it like the liquid that can leach into the soil can change the soil, which means you can have a very high concentration of copper in this soil. Now copper is a heavy metal. So anything that is growing in that soil should have the ability to resist the effects of this copper. So you might have some there. Now 50 feet down or 100 meters down away, where you have mining, non-mine soil, meaning there isn't any acid wash reaching there, there isn't any liquid leaching into that soil, there is no way there is a high concentration of copper there, meaning there is a lower concentration of copper there. You have a lot more lush grass growing. And when you check, there is no need for these plants in this population to have resistance to copper. And yet, when they kind of tested the grass on both sides, close to the mine and away from the mine, they kind of noted that there is a high index of copper tolerance in the mining soil, but there was also further down some population of grass still had the quality of mine copper tolerance further down, even though they were not even subject to that. Means in that environment they don't even have that stressor. And yet, how did this happen? Because of wind blowing and pollination happening crisscross. So in the mines also they had and away from the mines also they had a small percentage. This explains gene flow effect. Okay, so another example in this particular case where we have like a mountain range which is like a divider. So here you would have more golden colored deer and less dark colored deer, and away you might have darker colored deer and less golden colored deer, but there is a possibility of them crossing, and there is a possibility of these guys introducing their genes into this population and these guys introducing their genes into this populations, kind of evening it out, you might say. This is gene flow. Okay, then you have something known as relative fitness, meaning who decides who gets selected. Okay, the first and foremost in this, those who can survive with the changes being present and still mate and still leave offsprings are the ones that can increase in number, and they are the ones that get selected. But remember, selection can be a moving target, meaning the environment keeps changing. Okay, so if the environment keeps changing, that makes the adaptive evolution a continuous process. For example, a moth that is capable of producing more offspring than other moths in the same population because its body colors are more effectively concealing it from the predators, it improves its chances for surviving for long time to produce more offspring. Or you can have a barnacle that is more efficient in collecting food than its neighbors, may have greater energy stores of energy, and be able to produce a larger number of eggs, and so may able to be survive and pass their genes on to the next generation. Okay, so fitness, okay, it is, according to Charles Darwin, it is not the strongest species that survive, nor the most intelligent, but the ones that are most responsive to change. So, fitness from a selection point of view is a term that is used to measure how successful a specific genotype has been in surviving from one generation to the other. So, you can have absolute fitness or relative fitness. So, how does a trait change? So, first, what example I would like to use is there are different kinds of selection that can take place. Sometimes some selection would mean the environment changes in a certain direction, and so it is going to choose from one range to the other. Okay? On the other hand, or like one extreme to the other. On the other hand, sometimes instead of moving from one direction to the other, somewhat in the mid-range, the environment is choosing something that is against both extremes and somewhat in the middle. Okay. On the other hand, third kind is disruptive selection, wherein it does not like the mid form. It only prefers to have two extreme kinds being present in those two environments. So, examples or discussion of examples for each. Now, from one extreme trait, it can go to the other extreme trait. So, it selects one kind and totally rejects the other kind. For example, if there are lizards with short tails and long wiggly tails which look like a snake, that kind of seems to scare the predators. So, the longer the tail, the more it looks like a snake and the more it is able to survive in such a condition. And so, this is like a directional selection from having a short tail to a very long tail. On the other hand, a short tail can mess up a cat's balance. Long tails kind of drag it in the ground. So, medium tail seems to be the best for a cat for hunting and surviving. On the third example, extreme short tails can help keep the predators from catching squirrels on the ground, and extremely long tails are good for balance in the trees. Medium tails are not really helpful. So, we also have three possible outcomes in natural selection that is described just with one species of mice, but different colored shades. So, my point is, you can have a population wherein you have two extreme phenotypic variations, and original population had more of the medium colored or beige colored or dusty brown colored mice preferred. If the population changes from one form to the other, in the sense, now I kind of give the example to my class. Think about a mountainous region, open grassland, and some rocks. Okay. Now, mountainous cavernous regions, okay, would be beneficial for dark colored mice. Grassland would be beneficial for light colored mice. Rocky kind of partial shade, partial open can be favorable for in between kind of mice. Supposing in this environment all the kinds existed, and one day when we were trying to restore this area, okay, or there is a fire that burns up all of this grass, so that kind of selects from one kind to the other extreme kind. So darker colored may be preferred, or maybe having an advantage, selective advantage. Or another option of an environment wherein they kind of remove all these stones because they want to reclaim it and kind of construct it into a farming area or a grassland for more of the shepherds to use for their cows or goats or grassland animals, in which case the in-between colored fellows do not have an advantage. The darker ones and the lighter ones have an advantage. Now, third option is if we have bulldozed this mountainous area, removed all these rocks, and we are going to now have all of this, bulldozed this, and then also removed all the grassland area. Okay, so you're going to kind of have this clearing, in which case you might have the middle colored mice surviving. In any case, my point is, depending on what the environment is changing to or changing from, it can help select the specific variant which will be best suited for that environment that is present. So you can have directional shift, so you can have shift in the certain direction, or you can have a stabilizing effect, that is, there is no change in the sense there is no change from one extreme to the other, or you can have disruption of the existing range and new traits are coming up, or there is a loss of traits. Okay. This is again kind of running home the same point. Now, then you can have sexual selection. Sexual selection can be of two types: intrasexual selection and intersexual selection. Intrasexual selection is competition amongst individuals of one sex, often males, for mates of the opposite sex. Meaning two males will fight for the attention of a female. Like you can have two of these moose fighting for the attention of the cow. Or intersexual selection, wherein the individuals of one sex, usually the females, are choosy in selecting their mates. Okay, so the male's showiness due to mate choice can increase the male's chances of attracting a female while decreasing his chances of survival. Now, that can be explained a little further in the next slide. Okay, sometimes what happens is the males in any of the ecosystem, in the living system that you will observe, males are more showy and handsomer to look at than females. Females generally look very dull and drab. This is a question I generally pose for my class to ask them why they think so. And most of them invariably come up with the answer saying females are the one laying the eggs, hatching the eggs, and they are the ones they have more need to hide and stay camouflaged to escape the predators. On the other hand, males, because of them being showy, will end up selecting the female, but they are also like having a target on their back and like very easily detected by the predator. Okay, in some cases. Okay, so intersexual selection favors certain secondary sexual characteristics. The alleles for preferred traits increase due to non-random mating, and females prefer certain phenotypes. Like the female would prefer the prettiest male, or the female would prefer the most striking looking peacock, or in this case the pheasant. This leads to an increased sexual dimorphism. So here if you notice, peahen, though she looks very dull and drab, she always ends up choosing the best looking peacock who can show off his feathers. Now think about it. With longest feathers possible, he technically is very susceptible to attack by a tiger, or it's kind of cumbersome, and yet the peahen chooses him because when he opens up his fan and feathers, it looks like 1,000 eyes are looking, and it kind of scares the predators away. They also conducted an experiment to find out if frogs croak, how in the world does the female frog know which frog to get attracted or go towards. So they kind of caught two male frogs, one with a very high decibel, croaking decibel, okay, and one with a low decibel, croaking decibel, all right. And then they forced this lady frog to mate with this guy and this guy, and then they looked at the way the offsprings hatched and their pattern and everything. In that, what they noticed was the ones which were mated with the guy with the larger croak kind of had better larval growth, larval survival, and it had a shorter time to metamorphosis. So that was one way for the female to choose. Okay, you have a larger croak, so I like you and I'm going to mate with you. So my babies are going to be better. Okay, so this is her way of trying to detect it. But remember, it comes with a price. What is the price? Larger your croak, it can attract a female, so you can have a happy time, but it can also attract predators. Okay, so effects of inbreeding on variation. Non-random mating kind of puts a damper on this particular variations that can give you a better chance to survive life. Okay, so inbreeding, that is breeding with related individuals, will increase homozygosity, meaning you will have more recessive genes coming together, the chances of recessive genes coming together. How? Just think about it. So I would say, for example, polydactyly, okay? Not polydactyly, say disease X. Okay, I'm going to call that disease X once again. So disease X might be kind of like this. So if two people with one of the recessive alleles in them come together, they have 25% chances of having a baby with the two small d's and suffering from it. So you will have more of these deleterious recessive alleles that get expressed. This was observed in cheetahs when they kind of curbed their wandering territory, like migratory territory, or like their areas where they hunted. And then what happened was they started to inbreed, and so the babies had future generations of those babies. The chances of survival of those babies were decreasing because they had lower resistance to infection, and also there was decreased sperm motility. Okay, so one more question in this, like towards the end of this chapter. What is it that helps us maintain or preserve this genetic variation? First and foremost, the fact that we are diploid. We have two copies of everything. Okay, so if one is bad, we have the other. So then we have a chance of having one good copy. Heterozygotic advantage means even if you have like a bad copy, the good copy kind of helps you survive. Okay. Then there is something known as frequency dependent selection. I will come to that in a while. Okay. Heterozygotic advantage in a while in the next slide. There is also something called as neutral variation, which means there is variation in the alleles, but there is no selective advantage. Okay, but it exists, and As much as natural selection selecting the best fit, these are the forces that fight to keep up with the variation. Alright? So there will be some variations constantly being present which offer no specific advantage. Okay, so now we are talking about the heterozygosity. Now what I mean by this is, think about it, in these regions, okay, these are the regions where you have a lot of people suffering from malaria. Alright, now in this, these are the spots where people suffer from sickle cell anemia. Okay, now I would like to put the topic of sickle cell anemia first. Now, sickle cell anemia basically is, there is one what do you call, mutation and change in a base at the 6th base pair for the hemoglobin gene, and so you make a dysfunctional protein or hemoglobin, which kind of distorts the shape of the red blood cell. If the shape of the red blood cell is distorted like this, there is a host of other problems that can take place. One, they can block. They will not be able to supply oxygen adequately. They can cause a lot of other problems, which was discussed in our chapter on Mendelian genetics, and it was covered a few times before when we were talking about the structure of hemoglobin in chapter 5, and also during chapter on protein, gene to protein, you would have where 1 point mutation causing such deleterious effect, and sickle cell anemia being discussed as an example. But in this case, what I am talking about is the genetics of it. So who suffers from this? Okay, you have people, if they have 2 recessive alleles, they would end up suffering from it. So that means you can have people being SS, not at all having the sickle cell gene. You can have people kind of like a carrier for it, but not suffering for it. You can have people who suffer for it. Okay, now the advantage of this particular situation, the disadvantage, of this particular situation is you suffer from sickle cell anemia. But you do not die of malaria, because for this particular organism to set up malarial infection in your system, you have to have healthy red blood cells of normal shape. So people who have no sickle cell anemia gene would be more prone to die of malaria. Interestingly, people who have the sickle cell gene in them kind of live and do not die of malaria. So these areas are endemic for this particular disease, malaria. And so it is, though it seems like a cruel joke, being sickle celled will probably make you tired, make you sickly, but you will live. Then, frequency dependent selection is another option of selection. So for this, sometimes the fitness of a certain phenotype will decline because it becomes too common in the population, and then the selection favors the one that is not common. Okay, for this I want to explain about a certain lake Tanganyika. Okay, so there is this fish, okay, and this fish, has a mouth on, say, the left side. So this fish catches the prey from the back. This is the prey fish. If the fishes are swimming in a schooled pattern, it is able to bite them if they are all swimming to its left. Right? So what happens is, these prey fish are smart. When there is a high population of left-mouthed predators, they learn to smartly swim to its right. So they escape being eaten. Okay, so now what happens to this left-mouthed fellow? His number decreases. When his number decreases, the right-mouthed fellow, just 1 minute, where did it go? The right-mouthed fellow is able to catch this prey fish. So, I repeat, there is 2 types of fishes, predator fishes, left-mouthed and right-mouthed. Okay, if in the population on one of the seasons or year, I have a lot of right-mouthed fish. Okay, so let me just get back here. So say there is 1,000 predator fishes. Okay, and 800 of them are right-mouthed. So all of the prey species learn to swim on the left side. So what happens? This number decreases and the 200 left-mouthed fellows get ample food and they increase in number. So in the next season, out of the 1,000 predators, more number of them, say for example for our sake, more of them have become established in that population, which means 800 of them are left-mouthed, and now the prey population learns to swim onto the right side, which means they decrease in number and the right-mouthed fish increase in number, which is what this graph explains. So it says it is frequency dependent selection. Depending, if there is one particular kind of allele frequency that is in the population which is very common, it suddenly favors the allele frequency of the less common fellow, and it kind of goes as a zigzag, high, low, a high, a low, a high, a low, and so on. Okay. Finally, a discussion about why natural selection cannot give you something that is 100% perfect. Not 100% perfect. You cannot have that. The reason for this is selection can act only on existing variations or with helpful mutants. Meaning, with what I have to build. For example, I give my students an example. Supposing there is this man who is marooned in an island. Okay, so remember Tom Hanks, and he was like in one island, got stuck, and then he was trying to survive in that. Okay, so then finally he tries to put together with whatever material he has, he makes a boat, okay, or a raft. Which means what? You can make a raft with nails, good planks, you can have waterproof bottom, you can have sails with the best cloth. You can do all this, but do you have it? No. So I only have these trees, I only have this coir, I only have this leftover fiber, I only have this material from whatever parachute that fell off. So With that, I can build this raft. Similarly, my point is natural selection cannot fashion 100% perfect organisms because they can act with only what is there. They cannot create new. Okay? Then also evolution is limited by historical constraints. That means I cannot build something like a perfect creature from scratch. So if I have something, I can make a tad bit of a change to it and it can be different. A tad bit of a change to it and it can be different. So natural selection, like if you had this huge bulky land animal which later on became the precursor to a whale, which means they slowly, it had very short stubby legs, slowly its pelvis was very decreased, and slowly it didn't have any tail per se, it didn't have any limbs per se, and you will see the fossils like a gradually changing on to become the whale. All right, that is what was there in the slides when we were discussing about fossil records as one of the evidences given by Darwin in the previous chapter. Same way, my point is you cannot suddenly have a perfect creature that you can create. Okay, you have limited historical constraints. With what you have, you can make tad bit of changes and see if that works. If not, you make a change again and see if that works. Right? Adaptations are often compromises, meaning you will see in the next picture why. But before that, I would like to just talk to you about the black widow spider. Okay, the black widow spider, basically the female spider mates with the male and then eats him up. Now, why in the world would the male spider even come towards her? For him, his thought is at least my genes will be passed on into the next generation. So it's like a compromise for him. So either not get his genes passed on at all and stay alive but never get his genes passed on, or pass his genes on by the mating process and get eaten up in that process. Maybe he escapes it for one time, who knows. Or the next picture I have is this particular frog, if you notice, is croaking for its mate. Okay, but it is detected by the bat, so he would still croak. The point I'm trying to make is he would still croak. Okay, this is my frog and this is my bat. He would still croak and still try to find a mate. Okay, he may find a mate, he may pass his genes on, good, or he may be eaten by the bat. So it's like adaptations are often compromises. And then a lot of things come together to give you the final outcome, meaning there is always chance, natural selection, and the environment, like wind blowing and taking a certain log of wood containing a certain drowning creature to finally going and reaching a certain spot. So that's dry land and then they can continue to survive or not. Okay, I have two videos for my class to see at the end of it. Let's just watch that too. I'm done with this chapter, but I would like my class to see a video. All right, so let me see if I can pull that up. So the first one that I'm trying to bring in for you is, can you see a creature here? This is a mango tree. Okay, so that was one where I could show you from somewhere that I found a video which shows you that that particular caterpillar had completely blended to that leaf to be absolutely undetected by any predators in search of food, and it could continue to keep living. So this was not done overnight. That is my point. Okay, another video wherein you can look at different mechanisms of evolution as explained by this
variation is found in human populations and in other populations, such as this population of island beetles. Color in these beetles is determined by different versions of a gene, different alleles. Green is the dominant trait, so green beetles have two green alleles, or a green allele and a brown allele. Brown beetles have two brown alleles. By counting the brown alleles and green alleles, we can calculate that this population has 30% brown alleles and 70% green alleles. The frequency of brown alleles is greater than the frequency of brown beetles because some green beetles have a brown allele. Allele frequencies change as populations evolve. What causes populations to evolve? The island is suffering from a drought, and in this dry environment, brown beetles are better camouflaged than green beetles. Birds are more likely to see and eat green beetles. Over time, brown beetles survive and reproduce more than green beetles. This is an example of natural selection. Individuals with inherited traits better suited to their environment survive and produce more offspring than other individuals. Here, natural selection causes the frequency of the brown allele to increase. Through natural selection, the population has become better adapted to its new environment. Genetic drift occurs when populations evolve due to chance events. Let's see how genetic drift affects this small population of beetles. Chance constantly affects which beetles actually reproduce and which of their alleles they pass on. In a small population, chance events produce large fluctuations in allele frequencies from one generation to the next. Here, the frequency of the brown allele has decreased to 0, and the population is left with only green beetles. Genetic drift also occurs in large populations. But in a large population, genetic drift has less effect because it causes much smaller fluctuations in allele frequencies. Other random events can also lead to genetic drift. By chance, this volcano kills more brown beetles than green beetles, causing the frequency of the green allele to increase. Gene flow describes evolution due to individuals moving into or out of a population. When green and brown beetles migrate into the all-green population, the frequency of the brown allele increases. Natural selection, genetic drift, and gene flow are the major mechanisms responsible for the evolution of all populations. Together, they shape populations of beetles as well as humans.
And I hope that was helpful. I used videos from my instructor resources or from online to help my students understand, and I do not profit from this. It is just to make my students understand.